ఎస్ వాయిస్ వినిపిస్తా ఉంది అందరికి.
వినిపిస్తుంది సార్.
ఓకే,
వినిపిస్తుంది సార్.
ఎస్ ఇంకొకసారి కన్ఫర్మ్ చేయండి, వాయిస్ వినిపిస్తుందా?
ఎస్ సార్.
ఓకే, okay, good morning to all of you. We'll wait for another 2-3 minutes so that your other friends can also join the class. So in our last class we were discussing about enzyme properties of enzyme. Then we discussed about the nomenclature and classification of enzymes, and last class we discussed about enzyme action. So today we'll be discussing about enzyme inhibitors, then effect of cofactor on enzyme, and we'll be discussing about the uh, impact of factors responsible for enzyme activity. So just let me share the slide. So before going to that, I once again remind you all that you have to attend the online classes. Today morning also I have posted a press clipping from SK University stating that you must attend the classes. If you are falling short of attendance below 75%, then you will be in trouble. You will not be allowed for the examinations, and you will be failed. And in addition, you won't get any benefit provided by the government like the Jagananna Vidya Divana Vidya Vasati and so on. So be careful, don't remain absent, join the classes as and when the timings are given. Okay. Sarita Rani, you don't have, this is the class for second years. You join other group. N Sarita Rani, respond. N Sarita Rani.
Sir, I'm no mobile with me, sir. Yesterday I'm new mobile buy, sir.
ఎస్ అమ్మా, ఈరోజు క్లాస్ లేదు, ఇది సెకండ్ ఇయర్ క్లాసు. సో యూ లీవ్ ద మీటింగ్. ఈరోజు
Okay,
క్లాస్
sir.
లేదు. నేను వాట్సాప్ గ్రూప్ లో యాడ్ చేస్తాను కదా, దాన్ని బట్టి మీకు టైమింగ్స్ ఉంటాయి, దాన్ని బట్టి క్లాస్ అటెండ్ కండి.
Okay. Okay, sir.
ఇది సెకండ్ ఇయర్ క్లాసు, సో యూ జస్ట్ లీవ్ ద. Sarita Rani, log out Kandi, and Sarita Rani, leave the meeting.
Sir.
Okay. Yeah. So keep these things in mind and attend regularly. So let me share the screen. Yes. So I hope you are able to see the screen. The screen కనిపిస్తా ఉందా? ఎవరైనా రెస్పాండ్ కాండి.
కనిపిస్తుంది.
Well, we'll just quickly see what we discussed last time. So we were discussing about the enzyme action. For that, we have three models. One is the lock and key model. second one is the induced fit model. Third one is the Michaelis-Menten model. So in lock and key model, this was proposed by Emil Fischer in 1894, and this is just similar to why this is called lock and key model means every lock has a particular active site in it and a particular key suitable to that lock only. So that you can see here in this diagram that every enzyme has a particular active site where a particular substrate only can bind here. Just like a particular specific key for a specific lock, you have a specific enzyme for a specific substrate having a particular active site in it. So this was according to lock and key model. Then we saw the second model, that is the induced fit model, which was proposed by Daniel Koshland in the year 1959. According to this, enzymes they do not have a rigid fit but a flexible fit. They undergo some changes inside the enzyme for the perfect bonding to the substrate. They have two groups for this, so that changes, the active site changes that has to occur. The enzyme, the two groups that are responsible for that one is the dressing and catalytic group, because of which the changes occur in the enzyme according to substrate. That is how the enzyme substrate bonding occurs and the end products are formed. So it was that it was according to induced fit model. So we even discussed. So one thing here, Vishnu he has left. So I want to say one thing here. This is not that you have attended the meeting and class and you leave in between. Okay. So if you leave the class in between any time, then you will be treated as absent only. So don't leave the class till the end. Okay. So and sit in a proper place where the signal strength is good. Okay. So keep this thing in mind. If you leave the class in between, then you will be treated as absent. Yeah. So what was the basic difference between the lock and key and model and induced fit model was induced fit theory has had two advantages compared to lock and key. What was that? It explains how enzymes may exhibit broad specifics. So, example, lipase can bind to a variety of. Then the second one is it explains how enzyme catalysis occurs. The conformational change is this response in the substrate increasing the reactant. But in lock and key model, active site is single, whereas induced fit model is made up of two components, buttressing and catalytic. So the third theory, that was the Michaelis and Menten model, where this was proposed by Michaelis and Menten in the year 1913. So according to this model, the enzyme reversibly combines with substrate enzyme substrate complex that subsequently yields the product generated. So what is here, you can see here that enzyme combines with the substrate, forward reaction occurs, then the enzyme substrate complex is formed, and the enzyme and products are the enzyme and end product is formed. So enzyme is, so this is the forward reaction. So Michaelis Menten they uh, said that and the same reaction can occur in the backward, that is the reversible reaction can also occur in the presence of enzyme. So suppose the product is the substrate here and the enzyme combines, same enzyme combines here and the enzyme substrate complex is formed and the backward reaction occurs. Again you get the same substrate, that is the substrate which was used in the forward reaction. That same substrate can be regenerated as a product and the enzyme is released. So Michaelis and Menten they discussed on the reversible reaction. So they proposed that an uncatalyzed reaction, the number of substrate molecules in the transition state are less, so rate of reaction is low. And the presence of enzyme lowers the energy barrier, so more and more substrate molecules become reactive and react to produce end products, so increasing the rate of reaction. So here in this diagram you can see here, so this one is the this red line. Okay. This reaction is taking place without a without an enzyme. Okay. So here what is happening here? So suppose this is the uh, first stage where the reactants glucose and oxygen are the substrates. So here for this reaction, glucose and oxygen getting converted into products, that is carbon dioxide and water. Uh, it has to reach here, this point. Okay? So this point is the transition state or transformation state from where actually the substrates get changed into product. The actual reaction starts. So, so here this point, it has to reach this point. It it requires a lot of energy, activation energy. Okay, so here you can see here. So this much of activation energy is required without an enzyme. But when reaching this point, then only it starts changing to some product. But here, if you see the blue line, this one, this is with enzyme. Okay, so enzyme, what is it doing? It is lowering the activation drastically. So activation energy, the presence of enzyme is lowered very much. You can observe here. So the amount of energy required is lowered, activation energy, and the end product is formed at a very high pace. Okay, so you can easily see the difference here. Without an enzyme, lot of activation energy is required, but in presence of an enzyme, activation is lowered. That is how the substrates are getting converted into end products at a very high pace with the lowering of activation energy due to enzyme. So that was the point that was highlighted by the Achilles and Menten model. So overall, if you see the Lock and Key model, so it was based on the specificity, okay, substrate specificity. Right. Then the second thing was the induced fit model. It was basically uh, based on the conformational change that was taking place in the active site. Two components, that is buttressing catalytic sites, were there were present in the active site. And third one, mechanism model that was focusing on the lowering of activation, presence of enzyme. That was basically they highlighted the importance of the activation because of the presence of enzyme, how that affects. This was all about enzyme action models. Three models we have discussed. So now we'll be discussing about enzyme effects of cofactors on enzyme. Yeah. So here, if you see effects of cofactors on an enzyme. So what are these cofactors actually? So as we discussed in the properties, enzyme properties that enzymes are made wholly or partly with the protein component. So protein is very important component of an enzyme. So as the saying goes that in all, you can say that all enzymes are proteins, but not all proteins are enzymes. So Here, there are two types of enzymes. One is the simple enzyme, the one called as conjugated enzyme. So, based on the basis of chemical nature of enzymes, these two types are one is the simple enzyme and another one is the conjugated or hollow enzyme. So the first one, that is the simple enzymes, are made up of only proteins. Okay? You can say that simple enzymes are completely made up of only proteins and no other compounds. So the examples for simple enzymes are urease, lysozyme, pepsin, trypsin, and so on. So you can say that all these enzymes are purely made up of proteins only. And one more thing you can observe here, that during the nomenclature we said that every time based on the substrate or on the reaction, the enzymes have a suffix ase in it. You can observe here, urease. The enzyme is ending in suffix ase, but the exceptional cases are there where we don't add an suffix like the lysozyme, trypsin. Even though these are enzymes, we don't add ase. So there are a few exceptions for that nomenclature. So you think you remember that two types of enzymes are there based on the chemical nature, that is the chemical composition they are made up of. The one is the simple enzyme, another one is the conjugated or hollow enzymes. Simple enzymes, you can say they are made up of proteins. So examples for that is lysozyme. Second one is the conjugated or hollow enzyme. These enzymes consist of proteinaceous part called as apoenzyme, the non-protein part called as cofactors. So remember one thing, that simple enzyme is made up of only one component, that is the protein. But conjugated or hollow enzymes, these enzymes consist of two parts. One is the protein part as apoenzyme, which is made up of only protein, and the non-protein part is also attached to this part, which is made up, which is called as cofactors, which consist. These cofactors are what we'll discuss further. So remember, think that parts are there, one is protein part and non-protein. And the examples for conjugated enzyme or other enzymes are enzyme thrombokinase, then the salivary amylase. These are the examples for. Tarak, you want to say anything? In chat,
No, sir, by mistake,
yeah,
I have some.
okay. Yeah. So, enzymes, the examples for the conjugated enzymes are the enzyme thrombokinase and the salivary amylase. So these are the examples for conjugated enzymes. So here you can see here that holoenzyme has two parts. One is the apoenzyme plus the cofactor. Apoenzyme is the protein part, cofactor is the non-protein part. Coming down to cofactors, non-protein part of the holoenzyme, you can say them as activators. Okay. They are also activators. So cofactors are compounds or elements which help the enzyme in catalytic activity. So they are like activators of enzyme. As enzymes are catalysts, biocatalysts, okay, which acts as a catalyst during reaction, metabolic reaction, where the in this holoenzyme, the catalytic activity is activated by the presence of these compounds which are cofactors. As cofactors which are non-protein part. They are essential for the catalytic activity of holoenzyme. So this the holoenzyme, if it has to perform its activity, then compulsorily it has to have this two-factor part in it for its catalytic activity. So removal of two-factor causes loss of enzyme activity. If at all, by any reason, this cofactor uh, detaches from the holoenzyme, then the holoenzyme becomes inactive. So, definitely, these cofactors they have to be present. They act as activators. That is how the catalytic activity is activated in the holoenzyme. Then the cofactors may alter the shape of enzyme slightly, make the active site function. So this is like the induced fit feeding model. So the, there are some conformational changes at the active site for the enzyme substrate complex to occur. Same way, here cofactors they may alter the shape of the enzyme to some extent for making the active site functional in the enzyme. So here in this diagram, you can see here below that this is the apoenzyme part, the inactive part of an enzyme, you can say, which is made up of the protein, plus this is the coenzyme part, that is the cofactor part, that is the non-protein part of the uh, holoenzyme. So these two combine to form the one, the apoenzyme and cofactor. They combinedly form the holoenzyme. So here this is the holoenzyme where the apoenzyme part and the cofactor part they combine, and that is how here this region is the active site. Okay, so this active site is the point where the substrate comes and binds. So sometimes what happens here for the enzyme substrate complex to occur, this active site may undergo some slightly some change may occur, conformational change may occur in this non-protein part, two-factor part, so that the substrate part can attach here and the enzyme substrate complex can be formed. Okay, I hope you understand. Then the cofactors are of two types. Again, the non-protein part cofactors, they are further. There are some, you are not two types, there are some three types of cofactors are there, non-protein cofactors. So cofactors are divided into three categories. First category is the prosthetic group. So now, what is this prosthetic? All the three categories are, one is prosthetic group, second one is the coenzymes, third one is the metal ion cofactors. So first one, the prosthetic group are nothing but these are small organic groups, non-protein in nature, which tightly binds to the apoenzyme. So this prosthetic group are these are small organic groups which are non-protein in nature, and they are very tightly bond. They have a very strong bonding with the protein part, that is the apoenzyme. You cannot easily break down. So the example for this is the heme. Heme part is the prosthetic group of hemoglobin. In hemoglobin, the heme part is the non-protein part, which is tightly bound to the protein apoenzyme part, and it is broken down as part of active site of catalase enzyme. Second one is the core. So this is the prosthetic. Second is the coenzyme. Coenzyme are organic compounds, generally vitamin derivatives, which remain associated with the apoenzyme during catalysis of enzyme. act as carriers of chemical or atoms or electrons. So these coenzymes, they are different from prostatic group. These are also organic compounds only, but they contain the non-protein part like the white vitamins, derivatives, and so on, which remain associated with the coenzyme only during the catalysis action part of when the reaction occurs, catalytic reaction is performed by the enzyme, then only they remain active. They attach to the coenzyme part, the catalytic activity of the enzyme starts. Okay. So they act as just chemical carriers of the they act as the carriers of chemical groups. So that organic part may be a chemical group or it may be some atoms or some electrons. So for example for this is FAD, NAD, here NADH getting converted to NAD plus NH. Fine. So here flavin, adenine, dinucleotide, nicotine. So on all these uh, getting converted into NADH or FADH, where the broken breaking down of the electrons or protons occurs. All this is example for this coenzyme, the non-protein part. Perfect. Then the third one is the metal ions, who also called as inorganic cofactors. So the previous two were the non-protein part carrying organic organic compounds along with them. But here, metal ion cofactors are these are the inorganic cofactors. The metal ions bind to the coenzyme and participate in reaction by removing or adding electrons. So they are needed in trace amounts. The, for example, the peroxidase, ferrous, ferric, magnesium ions, and the hexokinase, etc. So metal ions are, you can say, they are called as inorganic cofactors. Uh, they actually, these metal ions, they bind to the protein part of the enzyme and they participate in the chemical reaction, metabolic reaction, either by adding electrons or by removing electrons. Uh, and they are needed at a very, very, very minute quantity. That is why call it as they are getting trace very negligible. So examples for this are peroxidase and the Fe2 plus ions, plus ions, plus okay. So remember one thing that cofactors non-protein part, they are of three types. They can be first one is the prosthetic group, which are uh, carry organic compounds along with them, which bind tightly to the protein part and the coenzyme. Then the second part are the coenzymes, which carry the electrons, atoms, chemical organic groups along with them. Third one are the metal ions, which are inorganic, which they add or remove electrons. They combine to the protein part and which are needed very, very less quantity. So, cofactors are compounds or elements which help the enzyme in catalytic action. They are like activators of enzyme. So, cofactors are divided into three categories: prosthetic, coenzyme, metal ions. So, these are small organic groups on protein chain which tightly binds to the coenzyme, whereas coenzyme they are the organic compounds and the vitamins and the minerals which are loosely bound to the enzyme. Third, metal ions, cofactors. So, so that was about the protein structure you can see. Okay. So remember one thing that these uh, enzymes mainly are of two types, simple and conjugated or normal enzymes. Simple enzymes are made up of only proteins, whereas conjugated or hollow enzymes made up of two components. Protein part is the uh, apoenzyme and the non-protein part just a minute. Yes, Sarita, respond, unmute చేసి మాట్లాడడం. Sarita, Sarita, unmute చేసి మాట్లాడు. So this is the class for second years. సో మీకు ఈరోజు క్లాస్ లేదు, ఆ క్లాస్ అయితే లేదు. సో యూ కెన్ లీవ్ ద క్లాస్. So here conjugated protein, they consist of the um, protein part, apoenzyme, and the non-protein part, cofactors. So the cofactors, as we discussed, there are three types: prosthetic, group, uh, then the coenzyme part, and the metal ion. So now we will discuss about enzyme. So enzyme inhibition. Inhibition means what? Blocking, to avoid inhibition. So enzyme inhibition here, you see, inhibitors are chemical compounds which decrease or stop the biological activity of enzymes by disturbing the normal interaction between enzyme substrate. So inhibitors are chemical compounds which either decrease or they completely stop the biological activity of enzyme, that is the catalytic activity of an enzyme. How they are stopping it or decreasing it means they disturb the normal interaction between enzyme substrate. So they block this activity in some way, and the enzyme substrate complex is not formed here. So that is how this thing occurs. The inhibitors they act. So inhibitors prevent the formation of enzyme substrate complex, hence prevent the formation of product. So the reaction won't occur at all. Okay. So here, if you see here, the so enzyme inhibition can be of three types. So inhibitors they stop or they block the enzyme activity, catalytic activity. The enzyme substrate complex is not that leads to non-formation product. Product is not formed. So ఇవి అడ్డుకుంటాను. అయితే enzyme inhibition ఇది మూడు రకాలుగా ఉండవచ్చు. There are three types. One is the competitive inhibition or reversible inhibition. Second one is the non-competitive or irreversible inhibition. And third one is the feedback inhibition. So these are the three types of inhibition uh, activities, enzyme inhibitors you can say, where they inhibit the enzyme activity. So we'll discuss one by one. So the first one is the competitive inhibition. In this, the inhibitor shows structural similarity to substrate molecules. So they compete with each other to bind at the active site of enzyme. That is the first one. So what is that denoting? That the substrate molecules which bind at the active sites the enzyme here. So this is the enzyme, and this region is the active site. So a substrate here, this is the substrate which has to combine, which come and bind here. But what happens here? And these are the end products. So what happens here is another another compound which is known as inhibitor, which is very much similar to the substrate structure, and that uh, uh, substrate structure, and which is very much similar to the active site. Yeah, that comes and binds here at the active site. So that uh, blocks the substrate entry. So when inhibitor binds to the active site, it forms stable enzyme inhibitor complex and reduce enzyme activity. So what is happening here? Instead of substrate, the competitive inhibitor, which is very much similar in substrate side, substrate as well as the active site, that comes and binds the active site. And what is happening here? It a stable enzyme inhibitor complex is formed, and which does not lead to the formation of product. No product is formed here. That is how the enzyme activity is reduced. As the inhibitor is in competition with the substrate, its effect can be reduced by increasing the substrate concentration, thus it is reversible. So, if at all we have to prevent this competitive, why we are calling this competitive inhibitor means it is competing with the substrate. It is same similar to substrate. That is why it is competing here, and that is combining with the enzyme. So, if at all we have to reduce this effect, then what we can do this here is what we can do here is we have to increase the substrate complex. Sorry, that's not substrate concentration. When the substrate concentration is increased, then only this reversible reaction occurs and the substrate enzyme complex forms. So this is with regard to competitive inhibition. So competitive inhibition example, if you see here first, succinic dehydrogenase enzyme catalyze oxidation of succinic acid to fumaric acid. Yes. So if malonic acid is added, the reaction, the activity of enzyme succinic dehydrogenase decreases rapidly as it structurally resembles the substrate succinic acid. Thus, malonic acid is competitive inhibitor of enzyme succinic dehydrogenase. So, succinic dehydrogenase enzyme, where it is having active site, which is uh, for succinic acid. Succinic acid is the substrate. So, what happens here is instead of succinic acid, if malonic acid is added, so malonic acid is also similar to succinic acid. This malonic acid acts as the competitive inhibitor to succinic acid that combines with the succinic dehydrogenase enzyme, whereby some changes occur in the enzyme and enzyme becomes inactive. Okay. Enzyme activity decreases, and this fumaric end product, fumaric, there is oxidation of succinic to fumaric acid, does not take place. Okay, this is one example for that inhibitor example. Another one is the sulfa drugs are used to treat bacterial infections as they work as competitive inhibitors inhibitors of folic acid synthesis in bacteria. Okay, so this is second example for competitive. Coming down to second type, that is the non-competitive. What are they? Sometimes inhibitor is not structurally similar to substrate molecule. So there is no competition for active site between them. So here what happens here is sometimes, as we discussed earlier, that these inhibitors, there may be some inhibitors which are very much similar to the substrate. They come and bind, they compete with the substrate, they come and bind in the active site of the enzyme and they block the enzyme substrate complex. So here in this second category, there are inhibitors which may not be structurally very much similar to the substrate molecule. So we call them as non-competitive inhibitors. Inhibitor molecule binding to a site other than the active site. We call them as allosteric site. So here in enzyme, there are two sites. So the site which is responsible for the substrate to bind, that site is known as active site. But there are some sites other than the active site where these type of inhibitor molecule they come and bind uh, to some other site other than the active site in enzyme. This type of site is known as allosteric site. So what happens if these inhibitor come and bind here? Then what type of action happens? Binding of the inhibitor to the allosteric site causes a conformational change to the enzyme's active site. As a result of this change, the active site and substrate no longer share specificity, meaning substrate cannot bind. Lastly, increasing the substrate does not affect the inhibitor. So this type of inhibition is irreversible. So what happens here is, first thing is that these are non-competitive inhibitors. Why non-competitive means they are not similar to the substrate shape or the active site shape. But in enzyme, these non-competitive inhibitors they come and bind to some other site the enzyme, which are known as allosteric site. What happens when these non-competitive inhibitors come and bind here? So when they bind to the that is the allosteric site, here the active site, whatever active site was there for a substrate, that active site they undergo some conformational changes and they totally are not similar to the substrate and the substrate cannot come and bind here. Just 1 second. So they come and bind here and that is how they block the enzyme substrate complex formation. But in previous case, what was that we can stop this thing. Okay, inhibition activity we can stop by increasing the substrate concentration, which was reversible. But when we could control the reaction. But here, what happens here is even though we increase the substrate concentration, that does not have any effect on the inhibitor. Thus, the inhibition is irreversible. Okay, we can't control it. So that is second type. So examples for non-competitive inhibitors are cyanide is a poison which prevents aerobic respiration and causes eventual death. Then second one, it binds to an allosteric site on the cytochrome oxidase enzyme, a carrier molecule that forms part of the electron transport chain. Then, by changing the shape of the active site, cytochrome oxidase can no longer pass electrons to the final acceptor, that is the oxygen. Consequently, the electron transport chain cannot continue to function and ATP is not produced via aerobic respiration. So, this is the example for non-competitive inhibition and feedback inhibition. I'm going fast because we have left with another. So, feedback inhibition is a type of allosteric or non-competitive inhibition which occurs in the biochemical pathway in order to control the amount of product formed in metabolic pathway. What is this feedback inhibition is? It is a type of allosteric or non-competitive. Maybe it can be either allosteric or it can be a non-competitive inhibition which occur in a biochemical pathway in order to control the amount of product. So this occurs when there is buildup of excess of product but the metabolic need declines. Cells use this method to maintain balance of homeostasis within the cell. The final product is a series of reactions. It inhibits an enzyme from an earlier step of this sequence. The product binds to an allosteric site and temporarily inactivates the enzyme by a non-competitive inhibition. As the enzyme can no longer function, the reaction sequence is altered and the rate of product formation is decreased. So what is happening here? So this is the beginning of metabolic pathway, intermediate products are formed, and here enzyme 1 is active combining with this intermediate product is formed, then another enzyme 2 is activating here, another intermediate product is formed, then enzyme 3 is here activating combining with this, and so what happens here is in feedback inhibition, the final product inhibits the enzyme. So what is happening here? In feedback inhibition, a type of allosteric inhibition occurring in the metabolic pathway of the living organism. The final product is in excess a series of reactions. It inhibits an enzyme from earlier step in the sequence and decreases the speed of reaction. So just I'm wrapping up this thing quickly. So enzyme inhibitors they block the enzyme substrate complex and do not form the product. So these are of three types: competitive, non-competitive, feedback. So competitive inhibitors they actually block the site for the substrate and enzyme substrate is not found, but non-competitive inhibitor they bind at the allosteric site, substrate becomes inactive, product is formed, and inhibition, feedback inhibition is an is type where allosteric inhibition that may occur or non-competitive inhibition may occur in metabolic pathways of living organisms. Okay? So these are the few types of enzyme inhibition. So I stop here. We'll discuss about factors responsible